Good morning or afternoon to you. Thank you for joining today's webcast. I'm Jan Reichert, Executive Director of the Antibody Society. As you might guess from our name, we serve as an international forum for facilitating discussion, disseminating information, and providing guidance on topics relating to antibodies. This is the fourth in a series of webinars designed to inform and educate our members and the broader scientific community about topics relating to antibody discovery and development. Our speaker today is Dr. Brian Kelly. He is the Senior Vice President of Process and Product Development at Vir Biotechnology. He will discuss delivering therapeutic mAbs for COVID-19. What can be done in just 1 year? Please note this webcast has been pre-recorded, but Dr. Kelly is standing by to respond to your questions. You may add the questions to the Q&A box in the viewer at any time during the presentation. Please watch the Q&A box then for responses to your questions. We will create a document based on the Q&A exchange, and a link to that document will be sent to all attendees. Thanks again for joining us today. Without further ado, I will turn the show over to Dr. Kelly.
Hi, this is Brian Kelly from Vir Biotechnology. How's everybody doing out there? Oh, wait, this isn't stand-up, it's a recording. Well, today I'd like to describe for you what has transpired in the last 12 months at Vir, working on therapeutic antibodies for COVID-19. It was a remarkable 12 months. Lots of twists and turns, and it took me and my coworkers to places we never thought we would go before. This is a disclaimer regarding forward-looking statements and other legal issues. Pretty interesting stuff, isn't it? In the talk today, I'd like to discuss speed to clinic objectives as well as speed to commercial, all at a pandemic pace, emphasizing the CMC considerations for all the development stages, and what has led to some very interesting overlaps in the early and late stages, as well as post-licensure life cycle decisions. This is a plot published daily by the New York Times of the COVID-19 pandemic and the time course of total cases in the world. It's just amazing as you look at the speed at which this virus and disease has been transmitted from March to August, resulting in over 19 million cases globally and over 700,000 deaths with a mortality rate that's greater than 3%. And the breadth of this disease, both by country and by the millions of patients that it has infected by any measure, is astounding. And yet there are many other examples of global pandemics and infectious diseases of significant impact. Vir is focused on treating and or preventing serious infectious diseases. And we list a few statistics for you on the right-hand side of this slide. Almost 2 billion people worldwide are infected with tuberculosis. 300 million people have chronic hepatitis B, and 37 million people are positive for HIV. In addition, we consider just in the US alone antibiotic resistance cases of 2 million per year, 250,000 hospitalizations for respiratory syncytial virus, and in some years, like 2017-18, almost 80,000 deaths from influenza. Now, there are some therapeutic antibodies licensed for infectious diseases, Synagis for RSV, Anthrax, and several antibodies that are now looking promising and probably will be licensed for treatment of Ebola. And so antibody treatment, one can consider prophylaxis or treatment of the disease. In prophylaxis, one has passive immunity granted almost immediately upon administration of the antibody. You could think of this as being important for frontline healthcare workers, for families of positive patients, and in the situation of an effective and safe prophylactic antibody, one would consider huge patient numbers globally to provide protection to the communities mentioned. In the treatment, one would be thinking of neutralization of the virus in vivo, and also the potential for T-cell engagement, depending on how the antibody is engineered and what characteristics or mechanism of action it may exhibit. In the treatment scenario, there would be smaller numbers of patients, but probably a larger dose than in prophylaxis. And so the patient number and the dose required might require an annual demand for an antibody for a single infectious disease of more than 5 to 10 metric tons produced per year. Now, in the COVID-19 world, we're seeking to produce those antibodies very rapidly, and I'll close the presentation today with a discussion of the production capacity and how we might approach this pandemic pace. If we think about influenza or other potentially pandemic infectious diseases that are known as opposed to being novel in the case of COVID-19, one could consider a stockpile of antibody in both the drug substance or the drug product configuration to enable rapid distribution in the case of a pandemic outbreak. Now, in addition to antibodies for treatment of infectious diseases, of course, there are many other multiple modalities. VIR is looking at several of them, and of course we have a whole host of vaccines in development today, and we hope that the combination of all these modalities, including monoclonal antibodies, will be effective in combating COVID-19. I'd now like to describe some of the methods used for discovery and molecular design of the antibodies. Some companies would approach using humanized mice or phage display libraries or other technologies. At VIR, the approach we take is described in the schematic. We isolate serum from patients who have survived serious infectious diseases, screen hundreds of millions of B cells from the serum and identify those rare antibodies with optimal characteristics. Then we engineer the antibody, either the Fab for developability and the FC for half-life and FC effector functions. Then we produce the antibody in very standard manufacturing technologies, and in treatment of patients, we transfer protective immunity upon antibody administration. Some considerations that come to play with regards to both discovery and then the dose that is required for treatment. First of all is the potency of the neutralization the antibody provides for the virus, but one has to consider, is the highest binding affinity always predictive of the lowest dose? I'm not sure that has been yet well established. Selection of the binding epitope of the antibody for the virus target is very important to seek highly conserved regions that would minimize the potential for escape mutants in the general population. Then for antibody design and modifications, developability we've all considered for antibody therapies of all sorts, eliminating chemical hotspots for deamidation, oxidation, potential for chain cleavage, etc. Half-life extension through tailored mutations in the FC can double or even triple antibody half-life and thus potentially reduce dose or frequency of administration. And lastly, considerations for up and down modulation of effector functions that might affect both mechanism of action and also mechanism of safety or toxicity. And lastly, considerations of whether a single antibody could be used for therapy or prophylaxis versus the need for a cocktail of 2 or more antibodies mixed together. The Ebola story is an interesting example of this. In the 2 successful antibody therapies that have been demonstrated useful in the Congo this year, one of them is a single antibody, the other is a cocktail of 3. So the dose implications of all of the characteristics or decisions above are huge. If we think about a situation where we're trying to produce such a large quantity of antibody, tons per year, the dose implications and production capacity are linked. And so a low dose or a single antibody versus a cocktail could have an effect of 2 to 5-fold on the total demand or supply that could be provided. Which brings us to the convergence of antibody CMC development. And we can ask ourselves, as David Byrne did, well, how did we get here? Well, the first recombinant antibody was licensed 23 years ago, Rituxan. And since then, there have been over 50 therapeutic antibodies that have been approved for commercial distribution and well over 1,000 that have been tested in the clinic. On the right-hand side, you see a common manufacturing platform that has emerged. The upstream and downstream unit operations are generally standardized across the industry, which has also led to design of manufacturing facilities to accommodate this platform process and general standardization across facility design as well. At the very bottom, there may be some recent advances to consider. Production at up to 25,000 liter bioreactor scale with titers now reaching 4 to 10 grams per liter. If you consider that with a 70 to 80% yield and debottlenecked downstream process train, one could conceive of batch sizes of up to 150 kilograms per batch. And that's a very different world than we were living in 23 years ago when Rituxan was licensed. But it's the capability that this provides that enables us to be considering these very large scale products for infectious disease. In addition to the convergence of the manufacturing process and facility design, there's been a convergence in timelines from gene to IND. In 2008, I went back to my notes from a benchmarking group with 6 major biopharma companies where each of them put their cards on the table and revealed what their timeline was from gene identification to filing the IND, and the range was about 18 to 22 months. So now, a little more than 10 years later, the convergence has been down to 12 months, in many innovator companies and CMOs, perhaps as fast as 10 to 11 months in some cases. And this was where my head was at in January when I was thinking about what the next antibody potentially to enter Vir's pipeline would be and how we would approach the phase 1 development, and predicting to myself it would take about 12 months to put that through. But then entered the COVID-19 pandemic. Well, let's take a look at what might be considered a fairly standard timeline for cell line development and process development for a phase 1 monoclonal antibody. In this 12-month period, which I think is pretty common for many companies who have practiced this for years and have refined their development strategies, we see the cell line development in various stages, including pool and clone screening, evaluation of cell line stability, and then testing of the master cell bank before entering GMP manufacturing. Using pooled material, we can initiate process and formulation development analytics as well. And in a fairly low-risk approach, consider using the clone to generate material for toxicology studies, which then become critical path to the IND, allowing the GMP drug substance and drug product, including testing, to be off critical path, and therefore there can be gaps between them and they're not needed to be scheduled immediately after the process lock. Just over a year ago, as the pandemic initiated, several companies decided to take antibody therapies forward to treat COVID-19. To move as fast as possible, several companies initiated first-in-human trials using material derived from a non-clonal cell pool. We'll refer to that process and product as the Gen 1 in this presentation. In parallel, we developed a Gen 2 process with clonal master cell bank, approximately 3 months later, material availability in the clinic. The key elements of the programs were to accept business risks, but not safety risks, to combine several recent advances that many companies have worked on, but perhaps not all have bundled into a single development strategy, to adhere to a strict platform manufacturing, no optimization, and find ourselves in the critical path in CMC when tox studies are shortened as a result of a viral target instead of a human target to be evaluated. So together with our partners WuXi Biologics, we put together the following strategy and initiated parallel development Gen 1 and Gen 2. The Gen 1 cell pool was generated in a traditional fashion using vectors and transfection selection that is used for clonal material. Transient cultures were used to fuel process, formulation, and analytical development. And as soon as a cell pool was selected with sufficient titer and product quality, it moved into GMP manufacturing with drug product scheduled immediately after drug substance. The cell pool was tested for adventitious agents by the standard panel used for MCB testing, but it was a business risk to initiate GMP production before all of the pool tests were completed. This resulted in a gene to IND timeline of under 5 months. In parallel, a Gen 2 process took a look at the top pool and cloned it to generate a master cell bank, which was subsequently tested. A platform check was run and used to confirm that the product quality coming from Gen 2 would match the product quality from Gen 1, and then the drug substance and product were scheduled after the MCB was available, but before testing was completed. Notably, the tox studies that came from the Gen 1 pool were supportive of the Gen 2 process, and so no additional tox studies were needed. Many companies began using cell pools for toxicology studies over 10 years ago, and now it is fairly common practice. We see that the Gen 1, Gen 2 strategy is essentially taking that pool for tox strategy one step forward. Note that the master cell bank shares the same pool genealogy, minimizing risks of significant product quality differences. The clone selection is biased for product comparability to the pool material. And in a pandemic, the speed to clinic really warrants use of the cell pool. We've mentioned the risks are not in patient safety, but really are in subsequent stages of development, as you'll see in a minute, when we need to prove comparability between Gen 1, Gen 2, and if later commercial lifecycle management strategies appear, an improved cell culture process for final production. This gives rise to a question: could this have broader implication beyond pandemics, but for other life-saving indications for monoclonal antibody therapies, where one is using a well-characterized therapeutic modality like an IgG1 antibody, and there may be a relatively small number of patients who are enrolled in a phase 1 study. Now, I'll use this timeline to depict the activities over a 12-month period, but for the first 5 months after lead identification in February, we initiated the transfection pool selection and process and formulation development as listed. Generating the Gen 1 pool material in the second quarter and employing a somewhat conservative drug product liquid formulation and presentation of 25 mg per ml, not having sufficient stability data to step into higher concentrations. And I just wanted to share with you some of the speed and the overlap of the program. There were 3 days in summer when we filed the phase 1 IND with the Gen 1 material on Thursday. The next day we had locked the Gen 2 process, which would be used for commercialization, took the weekend off, and came back on Monday to start designing process characterization and process validation studies, all before, of course, the first dose. These 3 days compressed activities in a fashion that was unprecedented for us. Next, we asked how fast can we file a BLA? A traditional timeline would have gated investments for phase 1, phase 3 development and production, process characterization, and process qualification, with a typical timeline of 5 years or longer from start to finish. So the key to achieving a rapid timeline is to put many activities in parallel. These would be conducted at risk, as we had mentioned, Gen 1 and Gen 2 being phase 1 and phase 3 development, running in parallel. We then stack process characterization on before we've manufactured all phase 3 materials, and work on transfer and process qualification while process characterization is still running. In the traditional timeline above, even a breakthrough therapy designation might not cause significant overlap. It would shrink the gating durations, though, and put them back to back. But running everything in parallel was a very different feel, and I'll describe a little more of that in a second. As I'd mentioned, the product comparability is key for all stages. We might be manufacturing at 3 distinct locations. Some companies have reported even more. And the ability to rest on structure function knowledge of IgG1s or antibody therapies is core to being able to prove comparability without a long history of manufacturing and product characterization. It's a bit like doing a biosimilar program at the same time you are preparing to launch, but without the history of a innovator company's years of manufacturing. And because of the rapid trial, speed to BLA is everything. CMC could easily be on the critical path to filing. Now, let's take a look at what happened in the first 9 months. After establishing the master cell bank, we locked the Gen 2 process after minor modifications to adjust and tune product quality attributes to match Gen 1. We increased the drug product concentration to 62.5 mg per mil based on additional stability data, and they became the basis for pivotal trial material. We conducted process characterization and validation in just under 4 months for drug substance manufacturing and initiated a tech transfer to a 6 by 2,000 liter facility, later running PPQ lots approximately 9 months after the initiation of the program. And I just want to highlight one of the unusual inversions here where I felt things were out of sequence in a way I'd never experienced before. We initiated the tech transfer before we'd even started process characterization and validation, giving you a sense of just how significant the overlap and the at-risk investment needs to be to make these very aggressive timelines work. Now I'd like to review the key development objectives and some of the decisions that they drive, with the goals of speed to clinic, speed to market, and capacity to supply paramount. Each company will approach their CMC challenges differently, and so therefore the timelines and outcomes will differ. Considering companies that have internal versus external assets, I'm living for the first time in a world where I'm completely outsourcing process development and manufacturing, different than my prior experience. The risk tolerance of the company for capital investment and technical risks. Clinical strategies that may have different timelines or multiple trials underway globally. And lastly, what feedback you get from the agency with regards to CMC strategies or approaches you've taken. Some of the key decisions I think of as where will one make tox material, in a pilot or in a GMP? In our case, we used a GMP lot because there was really no time advantage because of the very compressed Gen 1 process. In a Gen 2 process moving forward might just go for a pool for tox from pilot. The number of GMP batches, both the scale and number, this will be a key for clinical supply in the early months of the trial. Later come investments, in our case, of reserve capacity in very large-scale manufacturing facilities around the globe that comprise hundreds of millions of dollars of commitment. For a drug product configuration and storage, companies may generally lean toward their preferred platform, but that can vary significantly and have impact on shipping and supply for clinical programs. And lastly, something I'm coming to see now more clearly than ever, the control strategy, specifications, and evolution thereafter. All of these decisions I've just mentioned, plus a myriad more, were all needed to be settled within 9 months of the start. And we would skip from question and decision to question and decision, oftentimes feeling, I would say, not having a typical amount of information necessary to make a confident decision as I would have in a more traditional time. But instead, there was a feeling in the pit of my stomach many days that we just had to make a call and move on. Now, what has happened in the 12-month period, the last quarter for the program? We filed an IND for the Gen 2 process and drug product 2 at higher concentration. We initiated PPQ of the drug product manufacturing process, and I've indicated where we are roughly today in the dashed lines indicating that this activity is still taking place. We began working on a Gen 3 high titer process at the very beginning of the program, recognizing that this might take additional time for cell line development, including amplification and optimization of the production process. This is still underway today. We initiated a tech transfer 2 to the second CMO for a scale-up and additional commercial supply. And lastly, we've begun preparation for BLA to support the Gen 2 and DP2 processes. There are a couple more inversions that I observed in this period. The first is when we began PPQ runs of the Gen 2 process at the launch facility before we had filed the INDA for the Gen 2 process. So if the agency had anything that they didn't agree with, we would have to reboot the BPQs. In addition, we initiated Tech Transfer 2 to CMO at about the time before the INDA went in. This is an investment of hundreds of millions of dollars, again before agency feedback. So some periods of discomfort, but all in all, the ship is sailing as we speak. Finally, let's take a minute to look at CMC Regulatory Considerations. Not just accelerated filings in the US, but also global filings, some with 3 stages, including initial clinical filings, then emergency use authorization, and then hopefully licensure, all in rapid succession. It's a volume problem to be dealt with there, including many questions from different authorities. I'd mentioned the control strategy evolution. Keeping up with that in all areas and with different health authorities is also quite a workload. If we think about the process characterization and process validation chapter, one of the common questions for breakthrough therapy designations over the years has been, do we need the full package, or are there some studies that could be deferred? And generally the feedback we've gotten so far from health authorities is that they would expect the full package, similar to what we've heard for breakthrough therapies. We will have limited drug product stability at the time of filing, certainly less than ideal, and I'd love to hear some discussion in the meeting this week about how others may have approached that problem. And indeed, there's no time to recover from surprises or delays. And as mentioned a moment ago, a 4-month period for PCPV is extremely compressed for drug substance, but we were lucky to be able to see that there were no issues that arose. And lastly, can continuous process verification complement a relatively light process development experience in the post-licensure stage? But what is interesting is if the development database is relatively lean for these pandemic-paced programs, it turns out the full-scale manufacturing experience may actually be much larger than you would find for many other programs, by virtue of the need to build up significant launch volumes and have many more runs at full scale on the drug substance side than one would typically have with regards to standard timeline antibodies. And so with that, we're off to the races, as I know other members of the audience here are as well. Finally, I'd like to consider some production capacity limitations and ask the question, if 2 or more antibodies were approved for COVID-19 treatment, would each be capacity constrained? Would they be able to coexist? And it's my view that any number of commercial products will always be supply constrained, and that multiple antibodies will be used for treatment for global COVID-19 patients. I begin this argument with the Duke Margolis Center paper that was just published in June. They estimate that 40 million doses would be required in the US in the next 12 months. And if a dose were 0.5 to 1 gram, then it would be 20 to 40 tons per year, US only. And my estimate, just back of the envelope, for last year's global antibody demand for all blockbuster antibodies used in oncology, immunology, etc., is less than 15 tons per year. And at first blush, this might seem like an impossibility to satisfy a global demand when it outstrips the current production capacity from last year. But current technology enables a significant antibody supply. 10 gram per liter titers are now possible. And if produced at a 16,000 liter bioreactor with an 80% downstream process yield, that would be a 128 kilogram batch, and a 6-pack of 16K reactors could provide 20 tons a year. And there are many of these six-pack reactors around the globe, leading us to consider a question, is it time to think of a consortium? And so the second of the references on the right-hand column is just a little more than 2 weeks old. It comes from the Department of Justice, and it speaks to enabling 6 innovator companies working in the COVID-19 antibody space to discuss their manufacturing capacity plans and capabilities without infringing on any antitrust concerns that would typically prevent such conversation. I wouldn't be surprised if other companies join in the 6 that are listed there. And it's hard to predict what might come out of that, but it may be possible that there could be some benefit of companies with reserved capacity, but yet whose antibodies did not demonstrate efficacy, to be able to share that capacity with companies that have an efficacious antibody but are supply limited. And it actually harkens back, interestingly enough, to a paper published in Nature Biotechnology at the bottom of the column by Michael Kmark about almost 15 years ago. Michael Kmark was president of Wyeth Biopharma and Merck Bioventures, and now he is the chief technology officer here at Vir Biotechnology. And in that paper, he conceived the potential for a manufacturing build-out necessary to provide sufficient capacity for monoclonal antibody supply in 2006 through a consortium of multiple companies that would share the risk and the output of these billion-dollar drug substance manufacturing factories that each company could build, but some of which may then result in excess capacity, unused capacity. So perhaps everything old is new again. And with that, I'd like to summarize. The pandemic has triggered rapid development of antibody therapies in at least 6 large biopharmaceutical companies and many smaller companies as well. Speed to first in human trials has really seen a quantum leap forward from a 12-month traditional timeline and uses cell pools for GMP production in some scenarios. The clinical development may culminate in a filing within a year of the first in human trials, and it puts enormous pressure on CMC to meet that timeline and to present the full package of process knowledge and product characteristics. Lastly, I feel that sharing CMC and manufacturing experience and strategies in conferences like this could benefit more patients. So I'm very pleased to see that there are other presenters to follow who will also speak to the question of accelerating biopharmaceutical development in the context of antibodies used for COVID-19. And finally, we end up with maybe the broadest implications. Might there be a new future state where we would choose between accelerated and traditional pathways, maybe not Gen 1 and Gen 2 unless we're in a pandemic, but perhaps between a Gen 2 accelerated program and a more traditional 12-month timeline. Finally, I'd like to acknowledge the 3 main groups who have contributed significantly to the work shown here. The first is WuXi Biologics, where the process development and manufacturing were conducted last year, members listed to the right. The Vir Biotechnology co-workers, including Mike Kmark, who worked so tirelessly last year. And finally, acknowledging just a broad group of biopharmaceutical technical development leaders who over the years have contributed to our collective understanding in the community of large-scale antibody development and manufacturing. And in addition to WuXi, acknowledging Samsung Biologics and Biogen, who are working with us on the next chapter of developing highly productive processes to use at very large scale for supply potentially of several tons of antibody per year. And our partners GSK, who joined us last year in the development of these COVID antibodies. Finally, I'd like to thank you for your attention and be happy to answer any questions or have conversations during the panel discussion to follow, as long as my COVID beard doesn't scare anyone off.